And now, here we are at ESMO 2018, and we have SOLO-1. SOLO-1, I think, is an unprecedented study of frontline still, but using olaparib. So tell me about SOLO-1 and what it is.
So SOLO-1 is a trial that has been based upon really a lot of the other results of other trials in the recurrence setting that we're going to talk about, making use of this drug called the PARP inhibitor, which exploits cancer's underlying inability to repair DNA properly. It was a 2-to-1 randomization, olaparib versus placebo. So it was a placebo study, so it makes it even, the results more exciting. And patients received the olaparib and the doses 300 milligrams twice daily using the tablet formation. Patients had to have advanced cancer with a BRCA mutation associated with that cancer, deleterious BRCA mutation. Most of those patients were germline, but there are occasional somatic mutations that are found to be deleterious. And patients received, chemotherapy, evaluation by a gyn-onc surgeon, and I think that was another point I wanted to make, is that you talked about getting evaluated by a surgeon.
30% of patients in SOLO-1 had neoadjuvant chemotherapy.
But it's important to get evaluated by a gynecologic oncology surgeon. Chemotherapy or response to chemotherapy, and then are using the olaparib versus placebo as maintenance therapy.
So importantly, this was for BRCA-mutated patients,
Correct.
either somatic or germline.
Yep.
So you had to get that tested, really, to make the decision not to use BEV, to your point, so early on. So the hazard ratio is 0.30. It's interesting that the hazard ratio in SOLO-1, 1 meaning first line, and SOLO-2 meaning second line, was the same, 0.30. But tell us about the differences in the medians, and maybe although the hazard ratios are the same, which is kind of the way we talk scientifically, maybe the medians are so disparate that it's better to use a PARP inhibitor frontline rather than second line.
Well, it's sort of remarkable. So if you step back and look at SOLO-1, I don't think there's anybody in the field, and anybody at this meeting, are terribly shocked by the SOLO-1 results. I mean, we all anticipated it was going to be positive, because it is remarkable how the hazard ratios are so impressive and relatively uniform from SOLO-1, SOLO-2, NOVA, Ariel 3. So that's one take-home message. You could really be comfortable that at least in the BRCA patient population, you just have a tremendous activity for this drug. I think you bring up a very interesting point. What SOLO-1 will do is bring PARPs now into the first-line maintenance. There's just no issue about that.
Absolutely, agreed.
And I think it'll do that partly because of just the impact in the hazard ratio, but you're implying that you may actually get a bigger bang for your buck earlier on.
And
I have to add, vis-a-vis Ursula's comments, that the data for BEV may be the opposite, where if you look at the Ariel study, Aurelia study, you get a better hazard ratio than 218, but that's a separate issue.
Yeah, so think about it. So if the control arm in SOLO-2, second line, is about 5 months, you do a hazard ratio of 0.3, that turns into 19. You say, that's really good. 5 to 19 months, that's really good. But if the control arm is 14 months in front line, and you get a hazard ratio of 0.3, now all of a sudden you're 50 months. So 14 versus 50, 36 months, versus 5 versus 19, basically 14, 15 months. So that 36 months of PFS, unprecedented.
It's unprecedented.
Is it practice changing?
I do think it's practice changing. I think based upon the enormity of the results, I do think it's practice
36
changing.
36 months of PFS.
And I think, and then the other considerations to think about are, it's for 2 years. So
2 years of duration.
2 years of duration. The question's going to be, and again, that's just now going to be 2 years from now, what are patients going to do? Continue on, yes or no? I don't know about that.
But the clinical benefit persisted even when they stopped the olaparib.
That's correct. That's correct.
Right.
And the other issue is, and you just don't know this moving forward, but there are some women who, even with advanced cancer, who have underlying BRCA mutations associated with their cancer, they will be cured.
Do you think we're curing more patients here?
Well, it's
But
certainly
cured
possible on this.
without the use of olaparib, just with
I
chemotherapy.
see what
So you may be treating, but that's where I think you've got that sort of stopgap measure of having the 2 years.
But do you
in
think
there.
we're curing more with
Well, I
olaparib?
don't know that. We're going to see.
Could be. Don't know.
So I think one of the best
I
metrics
mean,
is
as you know, in study 19, which we'll get to later on, there are still patients.
11%,
Right.
and that's second line. 11% now at a follow-up of 7 years are still disease-free. So I like these landmark analysis. It kind of puts it in perspective. So you get 2 years of olaparib, and then you keep watching. And so at 3 years, a year later, 60% on the olaparib are progression-free versus 27%
Incredible.
on the placebo.
Yeah, that's
so
pretty amazing.
you've doubled the amount of patients that are progression-free a year after they stopped it. It's not like you stopped it and the cancer came back.
That's really amazing.
Right?
It's incredible. Yeah.
It's
They're amazing results.
They're amazing results.
They really are. Yeah.
Interesting. So the toxicity, there was only 11% discontinuation rate. The side effect profile was like SOLO-2 and olaparib generally. You know, marrow suppression, asthenia, and GI toxicity. Again, only 11% discontinuation rate. But there were 3 MDSs, and all 3 of those patients died, and all 3 of those patients had been on olaparib at least for a year. Does that concern you?
You know, I think
AMLs,
Yeah.
they all died. So MDS and then
as
progressed.
you both know, one of the initial impediments to getting FDA approval for a PARP inhibitor back in the ODAC meeting in 2014 was this underlying potential risk for AML or MDS. And I think what's been interesting is that in the subsequent 3 randomized phase 3 studies for maintenance and recurrence, so you mentioned SOLO-2, the NOVA study, as well as Ariel-3, and you look at PARP versus placebo, there really wasn't much difference in terms of patients with MDS or AML on one. It's pretty equal between both.
But here there's 3 versus 0.
But now you've got the upfront trial, and so it's 3 patients. It's never good, but these patients all have underlying, BRCA mutations, so they are probably going to be at higher risk just by the fact that they've got that BRCA mutation of having an underlying secondary leukemia. They've all had chemotherapy.
But it was 0 in the placebo arm.
No, I know. So it will always have to be a discussion with the patients, and it would be interesting to understand in those 3 patients what their counts were during. Was this something that was predicted
Yeah, I know.
during the
Excellent
chemotherapy?
question.
Yeah.